Oh. Oh man. All right. Is it going? Is everything going okay? So, I haven't done this in a while. Yeah, 6A, 6A and 6B. If you know, you know. If you don't know, well, we're going to be studying viruses, bacteria, basically all about them, but more about how they work in the next chapter, which is 6B, or the next topic rather. I've already gone through most of this topic, but now we're going to just review. I've reviewed this first page a bit more. But yeah, it's good to go through this again. There's still a lot of pages where I could use a second read through. So man, I've said this maybe like 200 times now. So first you talk about the structure and the roles of both bacteria and viruses, and I hear something. Why do I hear something? That kind of freaked me out. Hello? Hello? Don't do this. Okay, I keep on forgetting that I'm on poop. Oh, I forgot to say that. Oh, it's a campaign? I didn't know that. Looks great. Okay, one watching now, is that me? Who knows? Who knows? Okay. This is kind of what I've been up to. Oh man. Okay, so there are certain important structures and less important structures in bacteria. Well, not really important, less important, more like one is essential and the other one is like optional, or it depends on what where the bacteria is or what its function is. So first they act as the healthy things, so decomposers, and that can break down into like being natural flora by protecting skin or maintaining skin and gut, and helping cattle's like cows and stuff, ruminant animals, to digest cellulose, as well as acting as pathogens. So it's mainly two, one and two, and this just breaks down into two further. The structure is you have four main things. You have a cell wall, cell membrane, nucleoid, which is basically um, the suspended loops of DNA, and then cellulose ribosomes, and then you have your for movement pili. Sorry, that's not true. For movement it's flagella. Pili is for attachment, and for it's like surface antigens, right? So it's like yeah, attachment to host cells. That's what it's helpful for. Um, Slime capsule. Okay, this is this hides your antigens and protects from dehydration. It's gonna be like this thick, slippery substance around the outside of the cell wall. Mesosomes and plasmids. Obviously, plasmids is the extragenic material that codes for specific characteristics. We learned about how I'm pretty sure what was it? RNA. No, it was like lactose or something. And that was coded for a certain thingy in the plasmids. Um, I also remember like what was that thing? Plasmids contain this one thing which has three things like structural DNA, repressors and like repressors activators, as well as like one more other thing. I forgot what that was called. I'm not sure. Yeah. Anyways, mesosomes is going to be internal folding, and that's going to be, it could be the site of cellular respiration. It could also be the site of other things. We don't know actually what it's used for. Okay. Nobody knows. So viruses, on the other hand, they're a bit different. Um, first of all, their main role is going to be pathogenic. Like, no, it's going to be pathogenic parasites. Okay, so it's two parts: pathogenic parasites, or maybe you could even think of it as non-living, living pathogenic parasites. Okay, and maybe you can even add to that even more is saying small non-living living pathogenic parasites. Okay, and you can kind of list them all together in a way. Okay, and so how would we do that? First of all, they're pathogens. They always act as pathogens. They're always disease causing. Parasites meaning is that they need a host cell to survive, or they need a host cell to do basic like actions like reproduction or replication, and um, like evolving. I don't know what it's called, like changing their DNA frequencies, or rather allele frequencies and stuff like that. Like changing their genome, just developing their like character. Is a bit like that. They can only do that using the organelles, using the host cells apparatus, basically. And so they can only do that. If they can only perform these basic activities in a host cell, um, that makes them a parasite. Uh, Over here, they said they need a host cell for enzymes, organelles, and ATP for replication and protein synthesis. Okay, so yeah, development is protein synthesis, replication is reproduction, the basic actions of any animal ever, organism ever, and but they need a host cell for it, for their enzymes, ATP, and organelles, and so that's why they're considered parasitic. Now, why are they considered non-living living? Small is understandable, but why are they considered non-living or living? Well, since there are very few genes, right? We said that they are very small, so they or rather okay, that is never mind. It's classified as non-living because it's not composed of a cell. I mean, it's smaller than a cell. These viruses are particles, okay? And classified as living because although it can go inside another cell or a host cell to perform basic activities, at the end of the day, they still do it and they still behave like normal creatures. Like at the end of the day, like when you look at the results, you know, like it's still a living creature. You can't say it was non-living, right? Or else it wouldn't have done anything. Okay. Um. Okay. Cool. So it's about living and non-living, depending on where it is or what it's doing. Um. And it's considered very small because it has very few genes, and so it contains only a few types of proteins in its structure, and of course uses these host cells proteins like enzymes. um, you can think of the fact that like even enzymes could be larger than like the enzymes of a host cell could be larger than the thing than the virus itself. So that's how small it is. That's how few proteins it has to code for things like structure and stuff like that. So. Has anyone touched YTP ever since then? I haven't. Like, I definitely haven't. Okay, this week he did. Okay, but that was very one off. Okay, cool. Chugga 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 chugga. Let's turn off the AC for a bit. Oh, but I could draw these on. Yeah, I could probably do that pretty easily. But then again, um, I don't know. Okay. So yeah, that's how we explained it. A small non-living pathogenic parasite. So again, only disease causing, needs a host cell for its organelles, ATP, and enzymes to do protein synthesis and to replicate or reproduce. It can be considered non-living because it's not made up of a cell; it is a particle, and it cannot be considered living. I mean, it can be considered living once inside a host cell because it does do things like, um, it does do things like replication or reproduction as well as gene manipulation or gene. Oh, what's it called? Gene evolution. Yeah. Adapt. Like adaptations. Okay, fine. So it can do like stuff like adaptations as well, just like a living cell. And it's small because, well, we said it was a particle, but you can put that into a bigger. I mean, you can make that more understandable by saying that since it's so small, it can't fit even organelles or ribosomes or any enzymes in its, in its structure, which means that it can't do things like it can't have like it doesn't. That means it has like it doesn't really code for much, right? It doesn't code for much. It doesn't code for many structural proteins. It's pretty small in all things considered. So yeah, that's the kind of like size that we're talking about here. Um, Now, when you're talking about the structure, I like to think of it as like from inside to outside. Okay, so if we just drew like, do we have a drawing here? Does this work for us? Okay, let's simplify it further, and then we'll go back to that drawing. So okay, so um, internally, what we have over here, that's going to be your nucleic acids, right? That's going to be your double-stranded or single-stranded RNA or DNA, okay? All of this is going to be linear. Right after that is going to be your, no idea honestly, is it cell membrane? It has a lipid bilayer, which it gets from the cell membrane of the host cell, but that's an optional thing. Um, it has a capsmere. It has a capsid layer, protein covering kind of thingy. Maybe you can consider this the capsid layer. What would this be then? Or maybe I'm tripping and this doesn't exist. I don't know. Okay, here we go. Okay, yeah, so I am tripping. Okay, therefore we're gonna raise like, we're gonna raise this, I think. So this is a protein covering a protein layer, protein coat, that's what they call it, made up of capsids, or it's made up of capsids. But each capsid, I mean, like it's made, but it is called the capsid. It's called the capsid layer or the capsid coating. Um, just like some little information is that it's made from repeating units of proteins called capsomeres, and it's repeating so that it can conserve, or like the genetic material can be conserved to code for one protein many times, or a few proteins many times, rather than a whole string of them. That makes sense. Then we have the next really important one is going to be your VAPs, or your viral attachment proteins, particles, virus attachment particles. So obviously, I mean, it's in the name. It allows you to attach to the host cell to inject your genetic material, or to inject your viral genetic material inside. This is usually going to be like changed or whatever depending on the mode of attack of the specific, whether it's a DNA virus, RNA virus, double-stranded virus, single-stranded virus, all that stuff. Retrovirus as well. It depends, right? And so, yeah. Cool. That's pretty much it. Over here you can see the exact same thing. DNA inside, capsid layer on the outside. Right after that, oh yeah, we didn't talk about the lipid bilayer. So right after this, we have this lipid envelope, which is derived or taken away from, or like a bit of it is like taken away from the cell membrane of the host cell that it was in, and then right outside of that you have your receptors. Okay, your receptors, because these are the ones that are binding to the host cells, because the host cells have antigens or whatever or whatnot. Receptors, what is this called? I'm not very sure actually, but yeah, it's like it attaches onto that. I'm wondering, no, right? I would have to use the other one, which I'm not really sure what that is. Should any conscious creature draw a tear from the pond of sorrow, was the tear they shed not also yours? So true. I've been sitting for only 10 minutes. This is bad. I need that. It's okay. Um. Need someone to So we're gonna do 6A, 6B. I also wanted to try doing some math, like an hour of math. That's also, I feel like that should also be a priority in any case. But 1 second, I just want to take a picture of this and ask ChatGPT how I would use this. Maybe that's not a good one. Oh, I don't need to do that. I can just do it on my phone. Oops. We do one for weeks. Can you stop giving me these notifications? Who's this guy again? You're talking to me. Okay. Weirdo. Anyways. Who is this guy again? Just give me a minute. I want to just see how to do this. Okay. Oh, I'm not signed in. Odd. Okay, that's not gonna work, so Gemini it is. I'm not generating images. What? Okay. Um, Bio catch up is a part of a larger goal like ace my biology midterm. Maybe drawing finish and JPN piano book or like dedicate 5 hours a week to creative hobbies. Let's say it's October. Your goal could be establish consistent study routine for biology and practice piano 3 times a week. Calendar grid. Use this for deadlines, appointments, birthdays, major events. If you have a bio quiz, you write it on that date. End of month review. At the end of the month, you'll reflect here. Did you achieve your goal and what did you learn? Weekly view is creating the game plan. This is where you bridge monthly goals with your daily actions. You likely do this every Sunday evening. So, review of last week. Briefly reflect on what went well and what didn't. Priorities. What are the 5 most important things you need to accomplish this week to move towards your monthly goal? Example priority 1 complete biology chapters 1 and 2 catch up. Finish the front and side drawing project and this is the perfect place for recurring items for your list. Instead of writing them every day, you can make them to track. Habits to learn. This is the perfect place for recurring items from your list. Instead of writing them down every day, you make them official habits to track. Wait, really? Do you have a habit tracker? We don't. Oh, isn't just like tick them? This section is for multi-step tasks. Bio catch-up, including lab, is a great example of the project of the week. Daily view. This is the page you'll use every morning or the night before. It's where the specific items from your brain dump find a home. Spend a hypothetical day using our list. Goal for today. Choose one main thing. And then priorities. These are pulled from your must-do tasks, pulled from your weekly plan. Schedule. This is where you time block your day and fit in smaller. Before bed, quickly fill this out. Today's achievements, finish this, ways to improve. Got distracted during drawing, will put my phone in another room tomorrow. Using the system, your random list becomes a structured, goal-oriented plan. You ensure you're working on what's important, not just what's easy or urgent. Okay, yeah, so then I kind of messed up, didn't I? Because I mentioned a lot of goals for today. I guess the main one would be finishing 6A, 6B, because that would help a lot. Although, yeah, finishing bio syllabus would probably be the month's goal. Yeah, okay, so I'll write down the month's goal and today's thing a bit more better. Maybe I'll add on to the schedule as well. End of day review. Damn, this is actually really cool. I like this a lot. Okay, yeah. Oh, wait, sorry. Is it open? Who is this guy again? Oh, he never even responded. Like that's funny. That's great. I mean, he probably doesn't know me. It's okay. Fine. Maybe I'm. Yeah, I am just being a bit annoyed because it's like, what is this? You know? What do you mean? Who is this guy again? You can ask me. I'm like, I'm right there. Oh my god. I have these frogs, dude. I'm confused. Uh,
Charger, please. Did you just call me now? Eh? Have you lost the mic?
He called my mum to come downstairs.
Why are you calling her? Why are you calling him by his first name? It's not funny. Ruth, this is none of your business. Who do you think you are? What is wrong with you? You need to go back to Nigeria. One-way tickets. One way. This country has broken you. Seriously. I wonder how.
Then they bring my grandma downstairs to talk to me.
اكبر حقي اكبر ان شاء الله ما تطلعون جوازات سفر تدرسون تجون متعلمين ان شاء الله يا ابن الحلال ايش جاب سيره زواجها هيا ماني فاهم هذا ماني فاهم هذا يلا فهمي تفضل
I'll also get 3 loaded fries, spicy fries. I'll also get 3 chocolate shakes, please. Yeah, that'll be all.
وعليكم السلام الرجال مريض مريض يا قلبي سلم سلم على البنات يا رجال سلم بس وش فيك يا بحر بحر وعليكم السلام الرجال مريض.
If you could make one wish for the world, what would it be? I wish to close my eyes
I guess anyone knows that.
Yeah, ma, I'm gonna. What? So? What do you mean, bro? I'm not. Am I involved? I'm not
Astaghfirullah. God damn, man. Okay. Yeah, maybe I should keep my phone in another room. Dang. أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الله لا إله إلا هو الحي القيوم لا تأخذه سنة ولا نوم له ما في السماوات وما في الأرض من ذا الذي يشفع عنده إلا بإذنه يعلم ما بين أيديهم وما خلفهم ولا يحيطون بشيء من علمه إلا بما شاء وسع كرسيه السماوات والأرض ولا يؤوده حفظهما وهو العلي العظيم اللهم إني ظلمت نفسي ظلما كثيرا ولا يغفر الذنوب إلا أنت فاغفر لي مغفرة من عندك وارحمني إنك أنت الغفور الرحيم رب ارحمهما كما ربياني صغيرا ربي زدني علما ربي زدني علما ربي زدني علما لا إله إلا الله لا إله إلا الله. Okay. So after getting to know these four different structures of your viruses, now we can actually get to classifying them. Now we usually classify them by their genome and their mode of replication. So when we look at these three different types, okay, we have three different types. We're going to be looking at differences in, when we say mode, by the way, we see, I think mode of replication would mean frequency, right? What does mode mean? A way or manner in which something is done, right? So the way, the mode, which affects both the frequency as well as the method itself of replication is differed, as well as the genome, as in what kind of genetic material, how is it maintained and stuff like that, or how is it kept. So when we look at DNA, RNA, and retrovirus, we'll see that DNA, immediately you can tell it contains DNA, RNA contains RNA, and retrovirus is a type of RNA. However, although that they contain, or like if they're the same RNA, so that's one thing that they have similar, and so you know that they have a difference in the method or the mode of replication. Okay. Now, what's the first difference between RNA and DNA? It contains an RNA, and this one contains this one contains DNA, and this one contains RNA in its nucleic acids, right? Over here, we can also say that it is less harmful. Why? Because when you inject this DNA, when they inject the virus DNA, it makes up the provirus, right? And that will replicate itself as the host cell DNA replicates. Okay, so it's going to be replicating its DNA at the same pace or the same rate as the host cell itself. We can also say that the host cell is adapting and developing and doing protein synthesis and all that stuff, because we haven't halted anything like that. We're just saying that we're doing it at the same pace. Okay, I think that's true. We have examples: smallpox, adenovirus, bacteriophage. Okay, cool. And now when we talk about RNA virus, immediately we're saying it's a higher rate of replication, so it changes its antigen strain repeatedly. They don't produce DNA in the life cycle. Cool. So immediately you can say that if it's not, if it has a high rate of mutation, this is not really referring to high rate of replication, but I guess it could. Like genetic mutations happen where? During like meiosis, right? For eukaryotes, but like, I don't know. Like if we try to think of it as like something similar, where like during meiosis you're gonna have your variation or like mutation occurring. Maybe it's during replication, right? So the more that you replicate, the more mutation you'll have. Okay, and so maybe you can assume that the rate of replication is not like halted or is not limited by the rate of replication of the host cell in an RNA virus. And of course, so you can say high rate of replication means high rate of mutation, which means high rate of change in your antigen and your strain, which is how we kind of, this is the stuff that we use to like try to try to neutralize or stop an acid virus from attacking. By knowing its method, by knowing its antigens, by knowing its VAPs, by knowing its strain, we can that helps us be able to identify and be able to tackle it. However, if an RNA virus continuously changes that, it's hard for scientists to do that, right? Lastly, we have retrovirus, which is a type of RNA virus, but these guys have reverse transcriptase enzyme, which allows them to do some kind of special way of replicating. They have a complex life cycle, so vaccines are still being made for it. Cool. Um, you have HIV, you have Ebola, TMV, lambdaphage, and you have the differences in their structure, where you have different kinds of capsids, and you have whether they are enveloped by a lipid bilayer or not, right? So yeah, these are some examples. These are common MCQs. Now, life cycle of a virus, okay? So we're focusing again on viruses here. Now, two different routes of infection. We have lysogenic and lytic pathway, okay? So this involves with the mode of replication too, because as the like by the end of the infection, or what is infection, right? We define it over here. Pathogens being inside cells by evading barriers to entry. So if we say that it has two different routes to infection, that means it has two different ways. Let's say this is the host cell and these are the things. They have two different ways of getting into the cell, and after doing that, like then they can also do these other things like replicating and eventually taking over the cell and whatnot. So to be able to do that, there's two types of ways of doing that, basically, okay? And they do link together pretty well. So for the lysogenic pathway, we are immediately talking about virus DNA, okay? Or DNA viruses, not RNA, but DNA viruses, okay? It's going to be incorporated into the host cell DNA. The same way how we can like incorporate energy into a plant, we're incorporating viral DNA into host cell DNA to make a new kind of DNA, kind of a combination. We call that a provirus DNA. Now, when this provirus DNA replicates, both the host cell and the viral DNA will replicate. mRNA is not produced from the viral DNA because one of the viral genes code for a repressor protein which prevents transcription. So what's happening is that you're having this is your proviral DNA, but both are being replicated. However, when you're doing replication, maybe perhaps you're doing protein synthesis. However, protein synthesis does not occur because transcription doesn't occur, because as a result of this being replicated, it produces it codes for repressor proteins, which represses the transcription of forming any kind of mRNA, right? Okay. Can you get like cereal or something?
খাওনি কিছু না
No,
কেন
I don't know. Okay, করছে. Okay, cool. So you can say that there is no protein synthesis going on in the DNA of the, or rather in the host cell. Um, need someone to know. Oh, I've been studying with this break thing on. That's great. Can you listen to this? How does it sound? what's it called? Uh,
Is that Bandcamp?
mm-hmm.
Is that Bandcamp?
No. Thank you for the man. I want to show you that one video. Hold on. Ba ba Hey guys, welcome to another YouTube video. Today we're going to learn about Bio 6A and 6B. We're going to tell you the tips and the tricks and the licks and the flicks of how to get your maximum grades in this subject. First of all, you want to give me a subscribe and a like and a subscribe and a massive like and a massive vibe. Yeah. Thanks. Woohoo! Yay! Okay, sorry. Yay! Sorry. Okay, can we relax? Thank you. So yeah, virus DNA gets injected, virus DNA combine or gets incorporated with host DNA, makes provirus DNA. Now when host DNA replicates, viral DNA replicates, mRNA is not produced because transcription is repressed by the repressor protein made from one of the viral genes. Um, and the virus is said to be latent, as in it doesn't do any viral activities, it doesn't do anything what do you call it? It doesn't become, it's not virulent in this phase. And you're gonna call this period of time a lysogeny period, okay? Where the virus is part of the reproducing host cells but not causing an infection. In this area, maybe you can consider this as maybe the lag phase, right? Maybe. Either way, under these certain conditions, under some certain conditions, when the host cell is going to be damaged, then the virus in the lysogenic state are activated, right? So it's not going to be latent anymore when, under certain conditions, the host cell gets damaged, okay? So perhaps let's just make up a scenario when you've used enough of the host cell organelles, like, because we're not making mRNA, so at one point it's going to get damaged, right? There's not going to be protein synthesis, so at one point it's going to be like, yeah, I can't do this anymore, and it's going to crack. And when it cracks, then your virus is going to start activating and becoming virulent, right? And in this case, we're gonna consider that entering the lytic pathway. So at this point in time, when you activate your lysogenic state, we don't need to repress proteins anymore. So mRNA synthesis starts, and viral particles are produced, and the viruses enter a lytic pathway. So, oh wow, okay, I didn't think about that. So in this case, even viruses are not okay. Yeah, so it could definitely be the lytic. You could definitely consider that the lag phase, I believe. So you're not having like your virus is getting larger, but not necessarily is it increasing in the number. Do you understand? So, however, when the host cell gets damaged, and in that case, then the repressor protein decreases, and you're allowing like transcription of both the viral and the host cell DNA, right? And now that means you're allowing mRNA synthesis and viral particle synthesis. And so now viruses enter the lytic pathway. Okay, so what happens? Now you have loads of viral particles being made. What happens? The viral genetic material is replicated independently of the host DNA. So now it's not replicating just on its at the same pace. Now it's replicating on its own. Okay. Um, as the virus DNA is not combined with the host cell DNA, it will replicate independently. Okay, the first two steps I'm guessing is it's not like it enters the lytic pathway, so then it enters at the first step. That's not true, right? Because in this pathway, what happens? The starting beginning is different. We're not having any provirus because the genetic material of the DNA virus, or rather the virus, and the host cell DNA are not incorporated with each other. So as a result, you're going to be having independent replication of the viral DNA or the viral genetic material, allowing new virus particles to be synthesized and causing immediate lysis of the host cell and invade and like and by extension invading of other cells. So now over here, by the end of this, we're saying that the synthesis of viral particles is beginning. Over here, the synthesis of viral particles also begins. So you can imagine that by this point, it actually enters at the third step, where they cause immediate lysis of the host cell and invade other cells. Okay, so let's just try to diagram this through. Both of them enter the cell. However, now over here, we're gonna have a provirus DNA, and over here we're going to be having normal DNA and viral DNA. Okay. Now, this means that that means that transcription is not halted, which means immediate transcription means immediate synthesis of the virus particles, causing immediate lysis of the cell. Okay. Over here, after enough replication and like the lag phase goes on, and you have a more genetic material of your virus DNA, then we can go into the lytic cycle, where we can then do transcription of it and produce virus particles. Do you understand? We call this induction. I'm not sure. So what's the differences? Well, left is virulent from the start. Why is it considered virulent? Virulent is because immediately when it goes in, immediately when it gets injected, immediately when it gets injected, it immediately gets transcripted and immediately starts producing viral particles, which immediately causes the immediate lysis of the cell. However, in a lysogenic virus, this doesn't happen. It's latent at the start. There's no period of latency in the lytic pathway, but in lysogenic, there is. In lytic pathway, replication of viral DNA is independent. In lysogenic, it occurs as a result of being incorporated with the host cell DNA replication, and also there's no repressor protein in the lytic pathway, which is also why there is no latent period, you can imagine. Okay, third life cycle, however, there is a third one for specifically the retrovirus. Okay, so over here note how we have considered both of these virus DNA, not RNA. So we're not sure. I'm not sure how RNA is incorporated into this. Maybe RNA could do both, but maybe RNA only does lytic because we said that RNA has a mode of replication which is different as to lysogenic. Different as to DNA, where DNA, replicates itself at the same pace. So that immediately gives you the idea of lysogenic. And this one is high rate of mutation, which means high rate of reproduction, which immediately gives me the sense of, hey, this is probably following a lytic pathway most of the time. However, retroviruses, again, what was the retrovirus difference? The reverse transcriptase enzyme, right? It containing that allows it to do some certain activities, right? How viruses cause disease. So now that we know how it can cause it can do lytic effects, it can act virulent, how does this lead to a disease occurring, right? So the symptoms of disease is by mainly the lysis of host cells, causing host cells to release their own lysosomes and digest themselves from inside. That's one point. Or by the production of toxins that inhibit cell metabolism. So imagine we consider a disease something that hinders the basic functions or activities of a human or of an organism. Okay. What is the basic activities? Like development of the body as well as reproduction. Okay. Now, when we do lysis of the cell by releasing lysosomes from their own cells, destroying them, that reduces the number of host cells. That reduces the number of cells in the human body or in the organism's body, in the host's body. What does that mean? That means that you're having a lack of reproduction. Like you get what I mean, right? And then you're also having cell metabolism, which is development of the cell. If you are like hijacking organelles, you are, you know, not only are you like hindering the metabolism, I mean, you're going to kill the cell anyway, but also that you're producing toxins that inhibit cell metabolism. So those two factors being like halted or like hindered, that causes your disease. Okay. Oh, that's the end of the song.
Was the most obviously fake bullcrap I've ever seen, bro. She did not immediately start cursing him out and yelling. Um, by the 10-second point, she should have been like crying and yelling at the same time. What other naggers? Yeah, she should not be speaking English at all, except for saying the homeless hole like word. I don't think the guy that could word for that in Arabic. Yeah, no, this is stupid. And it's garbage, and you're stupid.
قاعد يسال المغرب عندنا شاي؟ عبودي you already let the homeless person in your house عبودي تقول له شاي 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 يشوي يشوي راسك اسمعي يا عبودي يا ولد يا عبودي طب لك انا 20 مره 15 تكسبسي why did you call me back call me back واخت خوايا جاتنا
Father, I swear to God.
Level 2, Level 3. Level 4 ein bisschen entspannt. Ja, Level 5. Handschuh. Level 6. Oh, scheiße. Einstieg. Handschuh. Licht dran. Est-ce que le stylo était autour de 50 mètres d'année? Ok.
I'm not that interested.
might say a dubious prospect. I don't know what you're saying. An immigrant exposed for his insufficient camouflage. Good afternoon, my fine gentlemen. How are you?
I'm well. How are you?
I'm also well. Wow, you seem rather articulate.
I appreciate that. You're very eloquent yourself.
Why, thank you, sir. Are you an American?
I am. Are you?
I am as well.
Wow, it's rare to meet an American that's so well spoken these days.
One might say a dubious prospect. I don't know what you're saying. An immigrant exposed for his insufficient camouflage. Good afternoon, my...
You from Morocco?
No,
From where?
London.
Originally.
Bangladesh.
Wait a minute. It's Mandala. Wait a minute.
No, it's called Bangladesh.
I said Mandala.
Yeah. There you go.
Okay, yeah, we're disabling reels. Yeah, this is getting embarrassingly bad. Oh dear. Another. Okay, no, it's been like 30 minutes, like 30 minutes ish. So yeah, decreasing the number of cells in your body and decreasing the cell metabolism of the body. That's mainly the biggest problem. Over here you have bacterial growth. So now we're looking back at bacteria from viruses, and we're looking at the frequency of their replication or the frequency of their growth, right? Now, remember when I was talking about the lag phase, and I mentioned it during the conversation about the latent period. That's a bit incorrect because the lag phase is there during bacterial growth, not really during viral growth. Now, I'm not sure what the difference between a bacteria growing and a virus growing. I haven't studied viral growth yet. However, bacterial growth is in this way where you have a lag phase where you're having them kind of just reproduce and get a little bit larger, so that then at one point then they can actually increase in size by a lot more. Now, of course, this is going to be a lot more different because you're not reproducing within the cell, right? You're just reproducing in general. These guys kind of do have the resources, have the organelles in order to reproduce like that, okay? But they do it in a bit of a different way. So these guys reproduce asexually, of course, right? And so, but instead of doing it by mitosis, they do it by binary fission, which is similar, okay? First of all, you want to know that it grows in size and organelles are going to be replicated. Then the time between cell divisions is known as the generation time. And this generation time becomes smaller after every single growth or every single replication. And so as a result, the bacterial growth is going to be exponential, okay? Um, so you have a formula for the exponential rate of the growth. We have log 10 and t. So the number of population, the log base 10 population at time t minus the log base 10 population at time 0, the initial population, divided by log base 2 times the time, okay? Not sure, I'm not sure if I understand that formula. However, using this, I'm guessing this is going to be a constant, but I'm not exactly sure. Okay. The growth curve is going to have 4 stages. So you're going to be having a lag phase where we're growing in size, we're adapting to the new environment and stuff like that. Log phase, exponential phase is when the rate of okay, now when we look at, like, we're going to be looking at abundance, right? And when we talk about growth or the rate of increase in abundance, then we're talking about the rate of reproduction, as opposed to, I mean, not as opposed to, but rather alongside the rate of death, the mortality rate, right? So when we say that the rate of reproduction is higher than the death rate, then we can surely say that it's going to be the population or the abundance is going to be increasing, and vice versa. So when we see log phase is, the period of time where rate of bacterial reproduction is as close to or at the theoretical maximum that it can get to, repeatedly doubling in a given period of time, you know that the death rate here is like the lowest, right? Now, the stationary is when you have the total growth rate as 0 due to, I don't know. So growth rate grows to a point where it's going to be equal to your. Sorry, the death rate is going to increase until it's equal to your growth rate, and now it's at a stationary phase. Then after the stationary phase, then you're going to be having your death phase, where your reproduction is going to be kind of overwhelmed by the death rate, and you're going to be having a decrease in population. Actually, well, no, death rate doesn't necessarily increase, but growth rate does fall. Like growth rate stops, and death rate increases continuously. So, so that's going to be into your essentially your entire life cycle or your growth curve of a bacterial colony over time. Do you understand? Check it, check it, check it. Now, why does the growth rate decrease, or why you could say why does the death rate increase, right? Now, no, well, that, but also mainly specifically why does the growth rate decrease at the death phase? That's the main reason, right? Um, and so that's going to be the number of nutrients decreasing, accumulation of waste products like CO2, which because all these bacteria are like maybe some of them are like yeast where they do anaerobic respiration and releasing CO2, lowering the pH, denaturing enzymes to cause death and no metabolism. So these are certain reasons why during the stationary phase and during the death phase, you're having no longer an increase in growth, right? This is very similar to the prey-predator cycle when we studied that. It's pretty similar, as well as the case study that we saw with the what's called the I don't know what it was called, but that cycle that we saw, where we mentioned that giving them food. Sorry, give me a second. Yeah. So it reminds me of the shoe hair thing, right? The snow hair thing, where it's like, even though, like, so you give it, we did two things. We gave it extra food, and we also stopped any predators from attacking it. So they were in this closed space with food, extra food, and we saw how their growth rate would change. And it resulted that this actually led to a very marginal increase in growth or abundance of the snow hairs or whatever, because they increased by a lot, but they also decreased by a really big amount. And this was because, as you increase your population, you're also going to, well, okay, increased by too much essentially. Because after increased by too much, then the competition for food was just too great, and that leads to like such a big decline. Do you understand? So if it wasn't as such a big increase, then it wouldn't have been such a big decline. So if you didn't actually close the predators, because then as it increased, you would have a controlled increase, right? Controlled in the sense that the decline wouldn't be as big as the incline would have been. So you would have had something like this. Because you're having animals controlling this increase the entire way through. It's kind of like this bottom up control. That's how AI explained it. I liked it. You have like a bottom up control and you have a top down control. So the bottom up control would have been um, like food, right? Like that's the thing. Like now that food is there, constantly being supplied, they never run out. And the top down control would be like so that they never exceed the amount of population to the point where competition becomes extremely like prevalent and supply is extremely scarce, right? It keeps it in this perfect range, which allows population to rise. And so that's what kind of this reminds me of, because you're giving it like, I mean, or rather, it's growing in this area full of nutrients as they're hardy creatures. I mean, they can survive through anything, but they they are in this abundant nutrient, broth or whatnot, and so they're going to be increasing like crazy until then it really comes back to bite them in the ass, basically, if that makes sense. Um, and so let's say we want to find out, you know, this growth rate, like calculate it or experimentally find it. The problem with doing that is first you need to follow microbial techniques, and then we can actually get into how it's being carried out. So this is how you culture bacteria, not just investigate, but culture them. And so first of all, there's two mediums by which you can do it. You can either do it by liquid or you can either do it by solid. Okay, so a solid agar or liquid broth, right? Aseptic techniques are not the same as microbial techniques. Microbial techniques deal with how bacteria can be grown and stored and experimented on, but aseptic techniques are especially they're included in the fact that they bring about how to safely and without how to safely kind of analyze and learn about these bacteria without contaminating anything, contaminating people, contaminating the sample, and maintaining fair results, right? And so following the aseptic techniques throughout the experiment, you can be able to culture bacteria in a way so that it's controlled, it's experimentally safe, I guess you could say, because the results you find are accurate. Like they're not going to be biased in any way. If that makes sense. Um, Yo, I'm scared, dog. Yo.
He's like, have you had an encounter with the devil? Yeah, I got both. The story is they took me all the way up this mountain. I'm on the top of the mountain. We finished the photo shoot. They had OD cars. They got a three-seater McLaren. They got a Ferrari. And I'm dancing. I was like, how y'all get all this money? How do you think? We do the money rituals. I'm glad. No, you don't, bro. How you get this money? Don't put me on. I'm trying to get like y'all. Glad now we do the money rituals. They're not I say, so what type of money rituals? They say, Illuminati never talk to you. But laughing. Still laughing at them. Like, you playing a game. You're like, bro, no, for real. Leonardo DiCaprio do all that initiation. I'm like, who is that? What did you talk? Okay, keep it PG-13. He was basically saying either you let them peg you or you do oral and you get whatever you want. If you want to be the next big actor, the next big whoever, I can take you to somebody. And all you gotta let them do is just do what they want to do. Just for a night. Just a game. Basically, that's how the industry really is. Take me to go get some don't. He said, we're not gonna take you get no food. Like, don't. We spend all this money to get you home. It's like this in front. You seen them play don't. Yeah, like Spider-Man. Yeah, from Spider-Man. Kind of like that. Probably this one. Fantasy. Like it's probably right. See somebody morph into something. Yeah, it's like that. It threw me all the way up. We in the 360 clearance. It's like a button you gotta push. He smack up mailbox. So now he hit the button and he get up out of there. Hit the button, he get up out of there. I see the button, I'm getting up out of there too.
Interesting. Thank you, Kaelan, for sharing. Yep. So yeah, you're using aseptic techniques. There's a list of those, but we're going into aseptic techniques again in a bit. Incubating temperature for it to grow. Maintaining culturing conditions, suitable pH, suitable duration, as well as anaerobic conditions prevented, like pH being controlled using a buffer, sealing petri dish with tape, but also allowing air to enter so that it's not an anaerobic environment. Here are some blah blah blah. Okay, so why are we culturing them, right? So above we discussed about how to culture bacteria, how to grow bacteria. So we're growing bacteria to grow the number of microorganisms so that you can see the effective antimicrobial substances can be easily seen. Now, in this case, yes, the effective antimicrobial substances. However, in another case, maybe it's to investigate the growth rate of a microorganism, right? How you culture? Again, culture in either solid egg or liquid broth. We're providing nutrients, conditions such as temperature, pH, and aerobic conditions. Selective media and indicator media. Selective media is going to be containing specific nutrients so that you're growing one type of microorganism. This allows you to have a pure culture, and you use an indicator media to identify the strain of microorganism as it causes certain species to change color. So here you're isolating this microorganism even further so that you can investigate that microorganism, because you can have an agar plate and it contains all the microorganisms, or you can have an agar plate which contains only one, a pure culture, and you do that by using selective and then indicative media. Now you have the aseptic techniques. What are aseptic techniques, right? We've already done these 1,000 times, but let's go again. You want to prevent contamination or entry of culture by other pathogenic organisms and of a person, as in you don't want the person to get contaminated or any pathogens or any lethal strain of a pathogenic bacteria or microbe to infect somebody else. This is why we do aseptic techniques. It's also, it keeps the effect of or the results of the investigation suitable, stable, reliable, right? Because it doesn't affect them, so you can use these properly. Okay, cool. Some examples are to work beside a Bunsen burner, using selective media, using indicator media, using sterilized equipment by autoclaving them, wearing gloves, cleaning your workspace with alcohol or minimizing the time that the culture is exposed to air, making sure that you open at a small angle, making sure that there's just a lot. Okay, usually most of this is going to be paper 3 stuff. Explaining their importance. So over here we're mentioning entry of microorganisms into the medium, which or into the agar plate or into the environment, the flora that we're trying to set up. The reason why we don't want this is because it competes with other cultures of microorganisms and reduces the growth, which reduces the results or the effects of the substance that we're trying to investigate. In this case, because in this case we're looking at the effect of antimicrobial substances. As well as the fact that people can also be contaminated and infected and develop the diseases if other microorganisms are by chance pathogenic. Lastly, what is infection? We've talked about how can a virus cause a disease, right? Causes a disease. How does a microorganism cause a disease? Because it gets into your cells. It gets into your cells and then does all that funk. But once it gets into your cells by bypassing, barriers to entry, then it's able to, essentially you are infected now, because now you have these pathogens inside your body. Okay. Now, here we talk about measuring growth of cultures. So now, see, measuring growth of cultures, that means you have already prepared your culture using what? Microbial techniques plus aseptic techniques, although it's included. So essentially, imagine you have the cultures ready, and instead of looking at the effect of the antimicrobial substances, you want to measure the growth rate, or you want to be able to measure the number of cells present in the bacteria culture at various time intervals to measure the population growth. Okay? So there's hemocytometer, which is counting cells. There's turbidity, which is estimating or qualitatively understanding or qualitatively, not necessarily qualitative, because you can have a absorption spectra, and then that becomes quantitative. However, it's called calibration curve, not absorption spectra. But you can have a calibration curve which allows a more quantitative analysis, but this is an optical method. Then you can have dilution plating, which is kind of similar to counting, except honestly I don't really remember quite well. 1 second. And also you have area and mass. So you have 4 different ways. Is this? No, there's more to it, right? Here, barriers to entry. You have 2 examples of viruses, and then you're done. You're done. Okay, cool. So, hemocytometer, I'm not gonna go into specifics because I don't really, I am interested in stuff like this, but I don't think I'm going to be really that interested in it until I actually get to do it. Anyway, so hemocytometer is kind of like you take from your sample. I'm sorry. You take a sample from your culture, you place it into this microscopic slide called your hemocytometer. It holds a standard volume of liquid, so then you count the number of cells. Well, rather, you stain that slide, and then you can count the number of cells which are alive. And you can do this maybe at various different time intervals, and seeing how the rate of increase in cells increases or decreases. What is the growth rate? What is the is it positive? Is it a positive correlation? Is it a negative correlation? Stuff like that. Optical methods like turbidity means that you're gonna be placing it in. I mean, usually this is gonna be for like liquid samples, right? Liquid broths and stuff like that. Not liquid broth. Liquid. Is it liquid broth? Oh, it is a liquid broth. So, yeah, for liquid broth, usually what you would do is you just having a. It's like an alternative way of measuring the number of cells by calculating or by analyzing the decrease in light transmission through the kind of mixture in which the bacteria grows in. So as the growth or the population of the bacteria increases, it becomes a lot more turbid, and so the transmission rate decreases. Now we can have a calibration curve for absorption or transmittance, which we can then use to estimate the number of actually quantitatively have an idea of how many bacteria were there or cells were there present in that sample at that time. And we have dilution plating, which is going to be a series of dilutions y. Okay, so when we have a sample of diluted culture, okay, so we take the culture and we dilute it, and we're spreading it on the nutrient-rich agar. Okay, so immediately, okay, so you have your main sample that we've made from the microbial techniques. Okay, from this main sample, you're going to be diluting it in series of times. Okay, now. You take the sample from the diluted culture onto an agar jelly, and then you count, they're going to be divided to form a colony, and you count the number of bacteria in that colony. And then using CFU and your dilution factor, or the point at which you diluted it, you can calculate the amount of bacteria in the original culture, this one. Okay? So remember, diluting the original culture, placing the diluted culture onto a onto a solid agar jelly, okay? Then counting the number of bacteria colonies that are formed, to then kind of reverse engineer back into finding the number of bacteria in the original culture, okay? And this assumes that each of the colonies on the agar has grown from a single microorganism, as to say that all of these things that we're counting are coming from one microorganism, the same microorganism in which these are coming from, which these are coming from, which these are coming from, which these are coming from. All of these are from one microorganism, okay? Or one microorganism is contributing to all of these, okay? No other one. Lastly, area and mass of fungi. So this time, instead of measuring maybe the number or the concentration, we're measuring the actual mass of the fungi. Now this is specific to fungi, right? And so if we incubate a culture of fungi with the same conditions at different temperatures, then you can see how the largest temperature, the temperature with the largest growth you can find as the optimum temperature. Um, why are we doing that? So, I mean, in this case, is to find out the optimum temperature, but in another case, maybe if you wanted to calculate the rate of increase or rate of growth of fungi, then you would do the same thing. You just let it grow in standard conditions. I'm saying standard, but it's controlled conditions. And if you control those conditions and then measure the mass or measure the area of the fungi that you're growing in every time interval, then you can kind of have an idea of what the growth rate would be. So over here, the area is measured by measuring the diameter of the fungal colony in the agar jelly, thingy, um, in the agar jelly culture. However, but and for mass, and so the largest diameter would have the largest area. This is basic stuff. Mass would be incubating the culture in liquid broth. Remember, this is at liquid. The other one was was on solid agar jelly. This is on liquid, and if we remove samples at time intervals, then separate the fungi from the liquid by centrifugation, then dry and measure until no more mass loss, and then we measure the dry mass of the fungi. That's going to be your way of finding out the mass change over time of a sample of of your fungi. I'm so bored, dude. Don't duck down, don't duck down, don't duck down.
Under Title 10 of the U.S. Code, the president has plenary authority. Has Stephen? Stephen? Hey, Stephen, can you hear me?
ISIS Chan was created by Japanese social media users in January as a part of the widespread online response to ISIS releasing a video threatening the lives of two Japanese ISIS Chan. The person of color, the person of color. can do. Ah, this guy's young. Definitely amazing. God damn it. Dirty cops. True jumps. No way, right? This is not that kid. The freaking shit. The tiny one. Thomas G. Sunil. Ho ho. Are we done talking about area and mass? I think so. There's nothing too difficult about it. It's just the only thing I need to really understand is that if I'm measuring the area, then you do it by measuring the area of or rather the diameter, radius of which the fungi grows. And for mass, you want to do it in a liquid broth, and then when you take a sample, you're going to remove the fungi from it by doing centrifugation, then drying and measuring, dry measure, dry measure, dry measure until you can't see any more mass loss. And that's going to be how you measure your mass. That's honestly the only important thing. So if I just rather, and then you just measure the diameter colony. Okay, so how do these spread? you can see 6 different ways, right? So, vectors are a living organism that transmits an infection from one host to another. Okay? So, if an insect has a pathogen and he puts that into you, that's considered a vector. If you get it from, let's say, intercourse, that's going to be a vector. The other person is going to be known as the vector. Okay? Now, inhalation would be droplets expelled from an infected respiratory tract by the other person. Another infection is established. So now we're getting into more specifics. So pathogens inside the droplets from the respiratory tract, which are ejected, and then and then you by chance receive them, okay, and you get infected. That is called inhalation. That is one of the methods. Ingestion is when food or something that you consume, like drinks, is contaminated by the pathogen already. However, most are killed while cooking. Formites are inanimate objects that carry pathogens from one host to another. So like maybe a rock or something, right? Direct contact would be skin diseases and sexual diseases. So, okay, direct contact and vectors are different because vectors are from a living organism that transmits the infection from one host to another. Insects would make sense. Direct contact is different, okay? Although maybe you can consider that the person which you like you get it via direct contact, however that person who originally had it is considered the vector, maybe. Inoculation is a pathogen, or maybe it's like insects, like some certain, no, not all, like it's not like mosquitoes, not all of them contain a pathogen, like malaria or whatever. Inoculation, a pathogen can be inoculated into the body directly through a break in the skin. So inoculation would be like contamination, right? You have the entering of a lethal pathogen through a break in the skin. That's going to be inoculation. So remember again, vectors is the person by which you get the or the organism or the person, a living organism or person by which this by which they spread the pathogen. This would be considered a vector. Second one is okay, what is it? A vector. Inhalation would be the droplets containing the pathogen from the respiratory tract of another person. Third one would be just like what I did right now. Third one would be, it's not inhalation, it's after inhalation it is, there's direct contact, where it's constant, there's 7 I believe, and there's 6. So there's direct contact, vector, inhalation, inoculation, non-living thing, forgot what that was called, and last one was ingestion. So food and then non-living, that's going to be called formites. Okay, those are going to be the rocks or whatnot that carry the bacteria, that carry the pathogens. Okay, so there's living, non-living, food, drink, droplet, contact, and inoculation. Okay, again, living, non-living, food, droplets, direct contact, and inoculation. Okay, living would be your vectors, non-living would be your formites, droplets would be your inhalation, food and drink would be your ingestion, direct contact would be your direct contact, and inoculation would be your inoculation. Now let's talk about some barriers to entry. So of course, the first thing that comes to mind as a barrier to entry for pathogens is our immune system. But that, I don't know. That's not written here for some reason. Maybe it is actually. So skin, that's gonna be your first. So now when you look at these 6 methods of spread, now you can kind of imagine what the barriers to entry would be for each of these 6, right? Inhalation would be maybe wearing a mask, maybe I don't know, not like covering your mouth when you sneeze, something like that. Ingestion would be cooking your food more thoroughly, or you know, being more antiseptic or being more clean, like maybe cleaner surfaces and stuff like that. But then when you say cleaner surfaces, then you can start to talk about formites, where it's like being more clean, being more hygienic allows you to have a more cleaner environment with less formites. Direct contact, you know, maintaining social distancing, stuff like that, which allows less stuff like skin diseases and stuff like that to happen. Inoculation, you know, wearing bandages, not waving your hand too much when you get cut, stuff like that prevents anything of the sort from happening where a pathogen could enter from your cut. Yeah. So skin going to be like skin is going to be a barrier to entry for inoculation, for example. That's going to be strengthened by proteins, structural proteins called keratin. It makes it impermeable. There's a lack of receptors, so nothing can bind onto it, right? Sebum is an oily substance that has antimicrobial properties and inhibits microbial growth. So that's also another thing on our skin. Skin flora, another bacteria, or rather, well, skin flora, it's going to be bacteria that grows there, and it prevents the growth of pathogenic bacteria by competing for space and nutrients and releasing substances that inhibit their growth. Maybe you can consider maybe it slightly being like a different pH, a different temperature. It's not optimum for them, right? Mucus lines the internal epithelial surfaces, and it is sticky to trap the microorganisms. And blood clotting seals wounds to prevent the entry of pathogens, like preventing inoculation. So again, you have skin in general made up of skin alongside sebum, alongside your what's it called, the oil. Skin flora, it's not oil, sebum is oil. Skin flora, which is the more of the bacteria, as well as the mucus inside the linings of the epithelial surfaces, as well as blood clotting. So if we try to draw a diagram to kind of help us understand that, let's say this is our skin. On top of our skin is sebum, and on top of everything is going to be your flora. Then below that is going to be your epithelial lining, which is going to be your mucus, and then at one area, let's say we have blood clotting going on, okay? So on the top we have skin flora. Skin flora allows bacteria to go there so that you're competing for space, you're competing for nutrients, and so and you're releasing, you know, chemicals that inhibit and bacteria or pathogen metabolism. Okay. The second one is going to be your sebum. Sebum is going to be an oily substance which it just helps to inhibit microbial growth the same way the flora does by having antimicrobial properties. Okay, cool. Then you have the main layer, which is skin, which is going to be an impermeable substance without any binding sites or receptors. It is made out of keratin, which is why it's impermeable. And then below that, we have these epithelial linings, which is kind of like the skin of, of certain organs or organelles. These epithelial linings are going to be lined up with mucus, so that it makes it sticky, so that microorganisms will just stick onto there. Pathogens possibly will not be able to metastasize, kind of, and travel. They'll be stuck onto one area until something can kill it, right? And then lastly, it's going to be the blood clotting, which prevents, inoculation prevents pathogens from entering the blood vessels or the blood vessels or the body in general. Now, when we are talking about chemical barriers, we're talking about microorganisms surpassing physical defenses. So now, let's say it surpassed all five of those physical defenses and is now in the body. Okay, what do we have in the body to help us? Okay, so we have saliva, which has bactericidal effects and salivary enzymes to destroy pathogens. Um, oops. So think about certain areas in your body. When we talked about saliva, that was ingestion. When we talk about stomach, that's also ingestion. Maybe saliva has to do some stuff about inhalation as well, right? You have your gut flora, which is the same thing as your stomach, I think, and vomiting. That's the same thing as just removing anything that you might have ingested, anything inside your throat, stuff like that, all being removed all in one go. So, in your mouth, first let's look. You have saliva. It has bactericidal effects, salivary enzymes to destroy pathogens. Lysozyme in mucus is also an example of a chemical defense. So we have those linings of mucus alongside your epithelial linings, and those contain lysozyme, which help as a chemical defense against pathogens. So the saliva in your mouth has bactericidal effects, as well as being able to destroy pathogens via salivary enzymes. Okay, so remember two things: salivary enzymes and saliva, bactericidal effects, and just being able to destroy enzymes. I mean pathogens. Now in your stomach, it has HCl acid, so low pH, so that it's going to be a bad condition for bacteria to grow. It also has low oxygen levels, so it's anaerobic, so not much respiration is going to be able to go on there, and so microbes growth is going to be halted or reduced. Gut flora, in addition to the gut flora, is also there to prevent the growth of pathogens in a similar way that the skin flora exists. It's going to be able to compete for space and nutrients because you're having loads of bacteria growing there, helpful bacteria. They secrete lactic acid to destroy pathogens, and they also have better adapted. The gut flora is better adapted to the gut conditions to outcompete them, right? Yeah, this makes total sense. And then lastly, you have vomiting, which removes microorganisms out of the body but results in dehydration. Okay, so now we've talked about how viruses can cause disease by reducing the abundance, as in by lysing, by causing the lysis of many cells. It's causing the lysis of many cells, as well as doing the other thing. What does it do alongside lysis? It halts metabolism by host cell metabolism by spreading toxins, right? So those are some certain things that they can do. Okay, so yeah, as those two things are halted or damaged in certain ways, that's why diseases are that's how we can consider them a disease, right? And that starts off because it gets infected, because the pathogens have a way of getting into the cell. Understandable. Now, how would we say that for bacteria, right? So, because bacteria, as we know, we don't know how they enter the cell. We don't know the method of their replication, nor their, other than the fact that they do it by binary fission, we don't know their replication, we don't know how they attack, none of it. All I know is that they have pili, but that even that's pili? Is it pili? What's pili pronunciation?
Pillis. Pilly. Pilly.
I call it pili. Pile. So, they invade host cells, producing toxins in their metabolism, which inhibit metabolism of infected cells. So, bacteria can enter host cells, produce toxins, which inhibit the metabolism. Very similar to how viruses do it. Let's see in a greater example how they do it. So they have endotoxins, which are lipopolysaccharides produced by gram-negative bacteria. They are part of these bacterial cell wall. They are part of the bacterial cell walls. They only has effect inside of infection. Once bacteria. Amor, I'm smelling something burning. Um, endotoxins, which are going to be your lipopolysaccharides. Remember when we talked about lipopolysaccharides? Not really. Where have I heard this before? Okay. So I don't know what lipopolysaccharides are. I don't know what gram-negative is. However, they're saying that it's in the cell wall, and since it's in the cell wall, it's only going to have an effect in the site of infection where that cell wall is being in contact. I guess. Once that bacterial cell is destroyed, the endotoxins are going to be released. So they're going to be released from the gram-negative bacteria part of the cell wall which was destroyed, and it's going to be released, and it's going to be released into the blood. The exotoxins are going to be proteins produced by both gram-positive and gram-negative bacteria, and they have greater effect as exotoxins are directly released into the bloodstream to have widespread effect. Okay. So they're not stored up or anything like that. Okay, where's my notebook? Oh, yo. Okay, so you have gram negative making the lipopolysaccharides in. Okay, so first of all, you can already spot some differences. Ay, ay, ay. Where endotoxins are going to be lipopolysaccharides made in the cell wall of gram negative, and when the gram negative bacteria is broken or killed or whatever, that's when you're going to release those endotoxins into the blood. Okay, and so since it's in the cell wall though, that the okay, apparently the site of infection is limited or something. I don't know how that works. Exotoxins, however, are usually proteins produced by both gram and first of all they're proteins and they're produced by both gram and gram positive and negative bacteria, and they are rather than being stored in the cell wall, they are going to rather than being part of the cell wall or stored in the cell wall, they're going to be directly just dumped into the bloodstream, and that leads to a widespread effect, causing a more like dangerous effect of the pathogen, similar to like how you had your DNA viruses being less effective or less dangerous than viruses and the retrograde viruses. Okay, retroviruses. Lastly, you have host tissue invasion, where bacteria invades and attacks a host cell directly. Then tissues by. Okay, sorry. Bacteria directly invades and attacks host cells. Then the tissues of all those host cells, usually by linking to endo and exotoxins, often linked to then tissues by often linked to endo and exotoxins damage the host cells, and then tissues produce the symptoms of a disease. Remember, it's going to be releasing and what these toxins do. Oh wait, often linked to endo and exotoxins. So they're going to be attacking host cells, usually by doing endo and exotoxins. This is going to be host. Okay, fine. So the bacteria directly invades and attacks the host cell, and then the tissue as a whole. Usually, this is going to be alongside exo and endotoxins. Now, understand that what these toxins do at the end of the day, we're saying that they attack them, we're saying that they're going to have an effect, we're saying that they're going to be released into the bloodstream, but what do? They're going to be able to inhibit metabolism of host cells. That's what they really are able to do. And when you have a lot of your cells being, their metabolisms being halted and stuff like that, when you have a lot of that happening, that's going to cause symptoms of your poopy. Okay. I just want to complete this so bad.
Tuberculosis.
I want to complete Please.
Okay.
Bro, I got the freaking exam virus, bro.
Tuberculosis.
Poopy virus. Well, you got tuberculosis. No, you got. What was the thing that you got? What was the thing that you got?
I don't know.
In-
Pneumonia?
Pneumonia. Yeah, that's a virus. It's honestly not that far from tuberculosis.
They say you can get double lung pneumonia.
I'm pretty sure it's very common to mistake tuberculosis for tuberculosis.
Oh, I think I remember
and
the doctor being like, oh, you have to check if you've got pneumonia or tuberculosis. And then they're like, oh, it's okay, you got pneumonia, you can get tuberculosis.
yeah, pneumonia is a lung infection that causes coughing, fever, difficulty breathing, etc. On the other hand, tuberculosis is another lung infection that can spread through tiny droplets. Ah, inhalation. Okay. Symptoms include this.
And then food.
Man, so many children die to like pneumonia. You know that? Like especially newborn children. Newborn children are so like what is the word for it? Vulnerable. There's another one, but yeah, vulnerable to tuberculosis. Apparently, there was like a Bangladeshi scientist which found a cure for babies who kept on getting tuberculosis, and then like the death rate like just declined by like crazy. What's the opposite of skyrocketed?
Drop.
That doesn't have the same effect.
Plummeted.
Yeah. Skyrocketed. Ground rocketed.
Ground flying.
Ground. Like
Ground falling.
like digging like really down. Like going.
Ground digging. Ground mining. Oh.
Like falling from like.
Ground drilling.
Like Burj Khalifa jumping. Meteor falling. Meteor crash. It was plane crashing. Oh, it crashed. Crashed is also another one. So, yeah, the amount of
Ground drilling.
the pneumonia cases crashed. Stock market crash. I'm crash. 2020 stock market crash. I suck a tire. Sorry. And then Google it. Wait. Hold on.
Chicken sandwich
Alright, I'll be back. Gotta go pray. Okay. We're quickly just going to get this over with. So, 3 ways, remember, recollecting from what we talked about, 3 ways your bacteria is going to cause disease. You're going to be, now it's quite similar to the way that we had one of the side effects of how viral, organisms caused diseases, where they produce toxins to reduce or to inhibit metabolism of your host cells. And it's kind of similar because these toxins also do the same thing, okay? But just how they're released is different, right? So you have endotoxins and exotoxins. Endotoxins are going to be they differ in the fact that endotoxins are lipopolysaccharides, where exotoxins are proteins. Endotoxins are only found in the cell walls of gram-negative bacteria, but exotoxins are proteins made from gram-positive and negative bacteria. Since it's in the cell wall, it's only going to be happening like in a smaller area for some reason, and this only gets released and gets used up when the bacterial cell is destroyed. Okay, remember these are in the cell walls of the bacterial cell. Okay, so the peptide dog glycan thing. Now, exotoxins, however, are released the moment they're made, so their effect is a lot more widespread, a lot more like instantaneous, kind of like the lytic effect as opposed to the lysogenic latent period, then when you have host tissue invasion, now this is direct like attack of the host cell by the bacteria, where they literally attack the host cell, directly invading them, and then furthermore the tissues by extension of it, by also using their endo and exotoxins to damage the host cells and produce the symptoms of the disease. And then we have 2 examples, and then I'm done. It's just these 2 pages, and then I'm done. It's so tiny, I can finish it in like 20 seconds. Okay, so we have 2 examples of viruses over here. We have tuberculosis and HIV. I don't want to really explain what HIV is in front of you, but tuberculosis I can do. তাই তো হবে, আমি তো সকালে গোসল করছি। I don't know.
সকালে গোসল করছিস, did you clean yourself?
Of course.
তাহলে বন্ধ হবে কেন?
কারণ আমি বাইরে বসে আছি।
তো তাহলে এসে আগে জামাটা চেঞ্জ করো।
Okay. Great job, Daisy. Okay, so we have method of spread. Now, it is a wrong idea. Okay, wait, is this a bacteria or is this a virus? Oh, it's a bacteria. Sorry. So since we already know it's a bacteria, we can already infer from one of these 6 methods of spreading. And for this one, it's going to be droplet infection, aka inhalation, right? The causative agent, we call this like the kind of vector, the bacteria, the name of the bacteria which actually causes this, because this is the name of the disease, or this is the name of the infection. However, the name of the bacteria which causes the infection is going to be called the Mycobacterium tuberculosis and Mycobacterium bovis. Okay, so these 2 things are a bit different, where Mycobacterium tuberculosis will infect people, and M. bovis will infect cattle animals, which then, when eaten, will affect people in the form of an ingestion kind of method of spread. Okay. Now, how does tuberculosis occur and result in death? So how it functions, essentially how it deals or how it goes on. Okay. So we have TB bacteria inhaled in lungs. Okay. Since we're talking about inhalation, droplet coming from the respiratory tract of one is going to be inhaled and going to be sent to the lungs. Now, when you're in the lungs, what's going to happen? So okay, let's just draw a diagram. Let's say this is one animal, this is one person, this is the other person. This guy coughs, and from his respiratory tract it goes in and it goes inside his nose. Okay, after it goes inside his nose, it's going to go into his lungs. Once it's in his lungs, what's going to happen? Okay, so TB bacteria is now in the lungs in the form of the droplet. Okay, and it's going to be engulfed by macrophages to form tubercles in the lungs. Okay, so I don't know what macrophages are, but macrophages exist to form tubercles in the lungs because macrophages cannot destroy them. Okay, so well, this is kind of like necessary now. I have to Google what macrophages are. Ba-da-ba-dum, ba-da-ba-da-ba-da-bum. Yeah. So what is macrophage? Learning about bacteria for the first time, really in depth for the first time, really. We're not talking about examples. So we have tuberculosis. The first step is it gets inhaled into the lungs. Then the bacteria is engulfed by macrophages. What are macrophages? Okie dokie. So, in simple terms, macrophages are a crucial type of white blood cell in the body that acts as a cleanup crew, security guards of your immune system. The name macrophage literally means big eater in Greek, which perfectly describes their primary function. Here's a breakdown of what they are, what they do, especially in the context of this. So, macrophages are specialized in. Okay, fine. That's all I really needed. So you have macrophages, which are like these immune system bodyguard guys, right? And they're there. I mean, of course, they're present in the lungs, and they are engulfed by the macrophages in the to form these tubercles. Tubercles, I guess you could call this complex, where it's inside this macrophage. Now, the macrophage can't destroy them. Okay, so what happens is the tubercles now result in this persistent coughing, which damages your gas exchange surfaces, right? Because you're having like rough contraction and relaxation of the lungs, and it's not good for you. So it's going to have some kind of damage on the gas exchange. Now, since the bacteria isn't removed, right? So maybe it's perhaps like this tubercle complex which causes this coughing, right? And so when you're having this breathlessness, this persistent coughing, this can result in ruptures of blood vessels, so systems of blood sputum in cough. Okay, so when you're having blood vessels being ruptured because of your coughing, the persistent coughing which is caused by the tubercles, you can see a symptom of seeing blood after you cough. Now, tuberculosis bacteria are whatever it's called, tuberculosis bacteria that were not destroyed, okay, as in which stayed in the form of tubercles, or I guess were not engulfed, remain dormant and reproduce when the immune system is going to be weakened to pass on their loss. So eventually they burst out of these tubercles. Okay, so what's going to happen is now it's inside this tubercle thing. It's going to be reproducing when the immune system is weakened. So it's going to keep on reproducing when they spot that the immune system is weakened. And they're going to pass on their lens. They're going to increase the allele frequency. They're going to do all these things, right? And eventually they're going to burst out the tubercles. And when they do, they're going to attack the T cells to stop antibody production and gets carried into the bloodstream to infect other organs, resulting in organ failure and other opportunistic infections can occur. Body temperature rises, but TB survives because high temp denatures metabolic enzymes. The adaptation that allows TB to survive past primary infection is that they produce this thick, waxy outer layer which protects them from the enzymes of the macrophages. Despite all these, TB cases are increasing worldwide due to increased migration of refugees, increasing intravenous drug use, and increasing number of HIV patients whom TB can attack as a secondary opportunistic disease. I don't know what an opportunistic disease is. What are you trying to do here? What is this? Uh, wow, well, that was just great, just absolutely great use of my time, wasn't it? uh, whew, okay. Yep, 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 yep. Okay. Yeah. Mm. While macrophages are designed to destroy bacteria, this one has evolved to not only survive but also to replicate inside the very macrophages that are meant to kill them. This turns the macrophage from a defender into a hiding place and a breeding ground for the bacteria. This is a central part of what makes tuberculosis such a difficult disease to treat. Is the battle between the macrophage trying to destroy the bacteria and the bacteria's ability to subvert these defenses is a key factor in whether a person develops an active TB infection. To try and contain the infection, your immune system will often form a structure called a granuloma, which is essentially a wall of immune cells, including many macrophages surrounding the infected cells. So from the ones which are so inside the tubercles, the TB bacteria which were not destroyed is going to reproduce until they're going to burst out the tubercles, and at this point the T cells can stop. They attack the T cells to stop antibody production, and then they get moved to other parts of the they get moved to other parts of the body through the bloodstream to infect other organs, and as a result, you can have organ failures. You can have other opportunistic infections occurring because at this point you're going to be having a pretty weak immune system, so other infections can come. This also means that your body temp is going to be rising, but however TB will survive. Now, how does TB survive past the first infection stage? Meaning, how does it survive when the macrophages come? They have a thick, waxy outer layer, which we know what this is, right? Which protects them from the enzymes of macrophages. Okay. Despite all these treatments and controls, what do? We diagnose using characteristic chest X-ray scans and analyzing bacterial DNA, mix of antibiotics to ensure all bacteria are effectively destroyed, so that if TB grows resistant to one, the other in the mixture will destroy it. Improving living standards, prevent going in crowded areas, prevent malnutrition of people with immune systems, drinking pasteurized milk, and preventing TB in cattle by vaccination, and then immunization, vaccination of people. But when you have increasing migration of refugees, as in, you're having people with one kind of, immune system or one kind of way of living, and you're mixing them with other people, maybe they're not used to living there, or maybe they don't have the immune system strong enough for another kind of ecosystem, then maybe it's possible for them to get infected like that. Anyways, finally we're talking about retrovirus, okay? And our key example of a retrovirus is going to be HIV. Now HIV it's not HIV, yeah. Now the causative agent of HIV is HIV, okay? So HIV, human immunodeficiency virus, okay, is a retrovirus. It's going to be an RNA virus having two strands of single-stranded RNA, okay? Two strands of a single strand of RNA. It contains reverse transcriptase, that's the key feature of retroviruses, and some other enzymes, okay? It contains a lipid envelope, and it has receptor proteins, okay? So they've told us already the main things. They have a few enzymes, okay? They have the protein, the lipid coating. I mean, like, they have a protein coating, of course, but they also have a lipid envelope or lipid membrane, whatever you want to call it. They have that, as well as surface VAPs, like GP120. Okay. Now, for this one, you're also going to be having a method of spread, and this is going to be direct exchange of body fluids. Okay. You're also going to be having it through inoculation, another very direct thing, and from mother to fetus at early stages of pregnancy, so this is also pretty direct as well. Now be clear about the terms. HIV is going to be a virus or a pathogen. AIDS, however, is a medical condition that's caused by an infection with HIV. AIDS comprises of a range of symptoms and conditions resulting from secondary infection by a number of opportunistic pathogens. HIV can be one of them. Okay? So think of HIV as an opportunistic virus. So when you have a body and that's gone through like a first infection, now it's like, okay, now be the third party, and come in. Now you're acting as an opportunistic virus, and that's what HIV is one of many, right? And the symptoms and the conditions resulting from these kind of opportunistic pathogens are called AIDS. Okay, cool. So now I learned about the three main ones again: direct contact, inoculation, and more direct contact. Over here we have how it's acting. So it's going to be binding with the receptor on the cell surface membrane. Okay, so they're both called receptors. Okay, cool. And then you're going to be injecting your genetic material as it fuses, as the cell membranes fuses. Then you can inject the inject the genetic material and the enzymes into it. So how does HIV really work? How does it lead to AIDS, and how does it lead to death? So okay, before we continue, let's just quickly re-explain this. So we have lungs, okay? Just gonna have lungs over here, okay? After droplets containing TB is inhaled and it goes to the lungs, it's going to be sitting there, and it's going to be engulfed by a macrophage. However, because of the waxy coating present, and also because mainly because of the waxy coating present in the bacteria, it's not going to be able to get destroyed in the macrophages. So what ends up happening is it turns into a complex called a tubercle. Now these tubercles can cause strenuous coughing. It can cause coughing, which can rupture blood vessels and lead to coughing blood or sputum, bloody sputum. Okay. And this can also lead to breathlessness. Now, the bacteria which survived within the macrophages, these will replicate until these will replicate at times of low immunity in the body, and after enough time has passed, it will rupture out of those macrophages. Now, immediately it will start attacking T cells to prevent the production of antibodies, and then also enter the bloodstream to go to other parts of the body, including other organs, to infect them. Once infected, now you're going to have stuff like organ failure. You're going to have stuff like being so immune, like weak in your immunity and in your like strength. You're going to be so weak that you're going to be very vulnerable to other opportunistic diseases. Okay, that's basically what TB is. Now, when we talk about HIV, it's a retrovirus and has these kind of VAPs, and they're going to be binding to the receptors of the T helper cell. So T helper cell. Now we said T cell for the previous one. So let's say there's a T helper cell, and it's going to be having these things, and we're going to be binding to them. Okay, cool. After binding to them, what happens? It's going to be ejected inside the genetic material, and the reverse transcriptase converts this viral RNA into DNA. Okay, converts the viral RNA into DNA. The viral DNA is incorporated into the host cell using enzyme integrase to form provirus DNA, and this provirus DNA takes over the biochemistry of the whole cell, synthesizing RNA, mRNA, and viral proteins to assemble into new virus particles. Protease is the third enzyme in HIV required for the production of these viral proteins. Okay, so immediately what the main difference that I'm seeing is that instead of just forming a lot of replicating a lot of the DNA, instead of doing that, what you're doing is you're by incorporating that viral DNA, what you've done is forming the provirus, right? As usual, but this provirus isn't going to just get read multiple times. No, what's going to happen here is that it's going to take over the biochemistry of the whole cell, which didn't happen before. What happened before was while it becomes provirus in the lysogenics phase, when the cell gets damaged enough, then the production of the repressor is going to be reduced, causing all the virus DNA to be read, which leads to it going into the lytic pathway. And then what ends up happening is, I don't remember. It's going to like do stuff. It's going to it's gonna like burst the cell membrane and like release lysosomes, which is going to like, you know, destroy the cell or something like that. So now you're synthesizing all these virus particles, and what they're going to do is they're going to immediately like break the host cell and invade other cells. So that's, yeah, okay, fine. So I was basically correct, right? It's just going to be replicating and immediately just breaking the cell apart and leaving. You're also going to be releasing releasing those lysosomes, which is going to digest the cells from inside, right? Okay, and now moving on over here. So when you're taking, so rather than doing that, rather than just getting your, you know, viral DNA read and then just synthesizing a bunch of virus particles and performing lysis, you're just taking control of the whole cell, including their DNA and their, you know, organelles and their enzymes and their ATP and everything, right? And using enzyme integrase. Now, first of all, do you remember the names of the enzymes? The first enzyme that was used to convert was called the reverse transcriptase enzyme, right? Now, over here, what we use to form the provirus DNA, to form the provirus DNA by incorporating the host cell DNA with the viral DNA is by using the integrase. Now, I don't know if integrase is used for the lysogenic one. I don't think so. Well, no, maybe. Yeah, no, maybe, probably. Because reverse transcriptase is the only one which wasn't present. So if integrase is present, then that explains how it's going to be able to form the provirus DNA. Now, this is a special kind of provirus DNA, however, because now you're actually taking control of the whole cell, acting as essentially the new nucleus, and you're synthesizing RNA, mRNA, viral proteins to assemble into new virus particles. So basically, you're now just basically a virus particle factory, okay? And protease is the third enzyme in HIV required for the production of, like, for this factory to occur, okay? No. Rather, protease is the enzyme that we're using to make the virus proteins, which are part of the. Am I out of battery? Which are present inside the virus. Host T helper cells are then destroyed as HIV, you know, increases in number. You know, kind of entering the lytic phase now. Now, as it's going to be released into the bloodstream, it kills the T helper cells, and T killer cells destroy the infected T helper cells, so the T cell count falls. Okay, so that's kind of how this is working. As a result, the immune system cannot fight other pathogens because it becomes weakened, so it's more vulnerable to opportunistic viruses or diseases like TB and pneumonia. Here we have some treatment and control. Presently, no treatment because it is a virus, right? The treatment by immunization, though vaccination is not yet available, because retrovirus like HIV has high mutation rates. So antigens change every year and take part in an evolutionary race. And so that's why we have to do immunization every single year or whatnot to ensure that, you know, this doesn't, so that we can always keep in track, you know? HIV weakens the immune system. Fewer T helper cells means that fewer B and T killer cells will be activated. And so TB becomes more prevalent. Treatment control, of course, there's no current treatment, but to prevent the spread, we have to do celibacy. That's funny. Oh man, I wasn't expecting that. Oh, that's actually really funny. Oh man. Oh, that's actually really funny. Oh shit. Okay, wait, give me a second. I gotta share that. who guard the sus among us? When you're cracking bro, but then he says some I love you. Ayo, that's so sweet. Okay. Okay, sorry about that. That is just really funny to me. Okay, so yeah, these things, I'm not gonna say these out loud. Of course, yeah, just prevent anything that reduces, as we said, these three factors will help you. So, I mean, this can't be done, but these two, yes, definitely, right? Making sure you're patching up your, you're patching up your wounds very quickly. You're making sure that the blood that you're maybe whatever it's called, centrifugation or whatever, the blood that you're taking in is going to be not infected, as well as the fact that the body fluids that is exchanged during the thing is not going to be infected with it either, right? Celibacy, only having one partner, using these, and using clean needles if injecting drugs, right? And then regular exercise to improve immune system. That's also very true, because we are seeing that the main problem with this HIV thing is that it results in a weak immune system, right? Because it ends up killing so many of the T helper and the T killer and the B and whatever cells, right? And so yeah, spreading awareness and education. Control is using antiretroviral drugs to delay onset of advanced stages of AIDS. Problem is that drugs are quite expensive. Yep. Okay, I'm done. See you later.